Next up, we have Thomas Pierrot, who will be telling us about the nucleotide transformer.
Thank you so much for the opportunity to present today. So I present the research that we're doing at InstaDeep and BioNTech in our group on building foundation models for genomics. And kind of to start this talk, I wanted to start with what is for us, at least, the definition of foundation models. Because I think there are still many definitions out there, and not everyone agree. And so I actually asked Gemini, because why not asking a foundation model for what is the definition for what it is? And what's fun is that it's kind of also the definition we had on our team and that you can find on Wikipedia and others. And so for us, we're going to go like foundation models, any large AI model that's been trained on lots of data and that can be used to like perform lots of different tasks. And so with that definition, we can take all the models trained with unsupervised learning or self-supervised learning that I present today, but also all the models that have been trained with supervised learning, for instance, to predict lots of different tasks, such as Borzoi or Informer, are going to satisfy this definition. And so I've put on that side lots of different sources of data that one can use to train these models, and lots of different models that have been trained so far, some of them that I cover today. And obviously, the list is much, much longer than that, as the field is kind of growing a lot right now. And in our group, the question we're asking is, how can we use all of this data, and even more, to build these models that eventually one day will maybe teach us new things about biology. And to start that journey, we started with these nucleotide transformer models, where the idea was to first learn from genomes and use all these techniques from NLP and unsupervised learning, saying, how can we take these genomes and run in a free unsupervised way? So we have no labels, we just have genomes from individuals, genomes from species, and we want to train a model that can learn something out of that and be useful. And the first idea we had, and that's now been done also in lots of other models and other fields in science, was say, okay, let's take these genomes. We can cut them into pieces. So for instance, in that case, we can take sequences of 12,000 kbps and then can kind of tokenize these sequences. So in our case, we decided to group the nucleotides by 6 to have 6 mers. There are lots of other choices that are possible. And we use a technique called mask language modeling or random masking to learn. So we mask 6 mers in the sequence, 15% for instance, and we train the model to reconstruct these mask positions. And we did that a lot on lots of data and investigate many effects. What happens if you just take the human reference genome? What happens if you take genomes from lots of different people? What happens if you add species? What happens if you make the model very big, smaller, etc. And kind of one of the first very cool results that kind of comforted us in the fact that it's interesting to try these approaches was if you, so in that slide I'm showing is the biggest model we trained on genomes from enough of different species. And after playing this game of reconstructing mask position in the sequence for a very long time, we took sequences for which we have labels. So our model has never been showed any label data. So it's just been shown sequences and need to reconstruct. We took sequences for which we have labels, so we know that some of them correspond to intergenic sequences, other to genes, UTRs, intron, exons, and so forth. We fed these sequences to the model. We looked at the activations at lots of different layers, and then we use UMAP or t-SNE, for instance, to visualize. And what was very interesting for us is that we observed that actually the model, despite the fact it's never been seen any labels during training, actually got these concepts of some of the genomics elements. But even more interestingly, if you look across the layers of the model, you can see that it learned these concepts at different levels of granularity through the layers. So at the very beginning of the network, it can make the difference between coding and non-coding regions. And if you go further, like it learns much more granular representation and can learn, for instance, to distinguish also UTRs. And we've done in the paper lots of these different experiments trying to understand what has been run by the model. And we show that, for instance, looking at activation, but also attention map, you can capture these patterns. And then the second set of experiments we did was say, can we now use these models as starting points to solve lots of downstream tasks? So can we fine-tune them? And this thing has also been done now a lot in the field. and in other fields, kind of what was important for us in that study was to try to find kind of a systematic way to evaluate design decisions. So how can we know, is it useful? So for instance, if we care about human data and predicting tasks in humans, is it useful to add the genomes of lots of people? Is it useful to add the genomes of other species, even if we care about humans? And so in our paper that's going to be published in Nature Methods in the coming weeks, we decided to build this data set where the idea was to cover lots of different tasks. So maybe not the harder tasks in genomics or the most important ones for a field, but trying really to cover lots of different aspects of the field and having enough tasks so that we can compute statistics and do robust analysis. And so by doing that, we were able to validate lots of design choices. And so, for instance, we show that, as in other fields, increasing the size of the model actually helps in terms of performance, but also adding lots of different species, for instance, in the training helps a lot. And so what's interesting also with that methodology is that actually now with the final version of the paper, we had to change all the data from all the tasks. So we still kept 18 tasks. We're still trying to cover the same effect, but we completely changed the assays and the sources of the data we used to compute this analysis. And despite that, we got the exact same ranking between all the methods and the exact same conclusions, which also show the robustness of the analysis. And since then, there's been many groups, including our group, but others also who run benchmark on these models and kind of kept showing the same types of conclusions. Also, a cool fact I wanted to mention to kind of conclude this section about the nucleotide transformer was we also asked, so in that idea of building models that can understand biology and not only DNA, we also asked the question, can we use these models to understand proteins? So in making the assumption that you have the coding sequence of the protein. This has been done already by people trying to build models based on codons. In that case, people train data set of CDS or of mRNAs. But in our case, we're OK, our models have been trained on full genomes. So for instance, for humans, it's only 2% of coding sequences. There are introns between the exons, so the data distribution is pretty different from coding sequences. So we just asked the question, how these models are going to perform, and we compared, so we took hours or so DNA birth, and we compared to some models for proteins, including ASM. And we saw that on some tasks, as we could expect, protein language models are much better, especially on tasks where you need structural information. But what's interesting is that actually, and as it's been showed for codon models, on some other tasks, the genomics language model actually even outperform the protein language model. But more interestingly, that we show that these two models are actually additive. So if for a protein task you have access to the true CDS and the amino acid sequence, you can take a model trained on genomes, can take a model trained on amino acid sequences, and combine them, even in a very simple way, and just fine-tune this model end-to-end. And you show that then on all of the tasks of the study, at least you get the best performance of both worlds, but in some cases, you even get additive transfer. And so, for instance, for protein stability prediction, you even kind of get state-of-the-art performance by combining the two types of approaches. So now for a second part of this presentation, we'll show how we started from building these models with self-supervised learning and what are the types of things we can do with it. And so I first present quickly our SegmentNT approach, and then show also ChatNT, which is a very different type of idea. And in that SegmentNT work, kind of the motivation to start this study, we say, OK, for now, we mainly looked at this problem of understanding genomics as an NLP problem. So, OK, DNA is a language, so we could use techniques that have been developed for language to understand it. But why not also looking at this problem as a computer vision problem? So in computer vision, people now also build very powerful foundation models. I'm thinking, for instance, about SAM, a segment anything model from Facebook that's kind of understanding the world by segmenting objects inside. And so when you look at genomics, so it's not going to be a genomic sequence, not going to be a 2D world, but a 1D world, but kind of similar to a computer vision problem. You have a background, and on top of this background, you have elements. Some are bigger, some are smaller, and they're going to interact together at different levels. And so it made us ask the question, could we build, similar to what's being done in computer vision, a segmentation model that's going to run to segment these DNA sequences and kind of teach us genomics through that lens. So that's what we did in that study. So we decided to start to focus on 14 elements, some regulatory elements, some genic elements, and we've built this simple system where we take our nucleotide transformer that has been pre-trained using supervised learning, and on top we simply plug a UNet, which is kind of the state-of-the-art architecture for segmentation. It's been used for a while now in many applications, and we train that model end-to-end on all these annotations. So for one DNA sequence as input, we're going to predict at each nucleotide in the sequence 14 numbers, which correspond to 14 probabilities of belonging to the elements. In that case, they don't need to add to 1. Each number is going to be a probability between 0 and 1, so you can belong to several elements at the same time. The first interesting result was actually this task is not easy to predict. So if you just try the UNet directly, you can get to some performance, but actually there are many elements you don't manage to predict. Then you can make it bigger. You can also take the architecture with lots of transformer layer and stuff and try to predict directly. But what we show is that if you actually do this self-supervised learning on lots of genomes, the performance gets much better. So you already get some transfer from learning on genomes and then transferring to this annotation. Another interesting fact was there's been many models in the nucleotide transformer, in DNA BERT, in other papers developed to predict if elements, for instance, are unanswered or promoters as a classification task. So you give the sequence and it tells you yes, no. What we show is that if you actually take these models also and slide them over the genome, the performance degrades a lot. And it actually works much better to actually directly treat the problem as segmentation and predict it this way. Another interesting fact was you can also get transfer learning on the sequence lengths. So we did this curriculum learning where we first trained on sequences only of 3 kbp. So we took the genome with all this annotation, chopped it in 3 kbp sequences, trained, and then we started the training again on 10 kbp, 20, 30. We stopped at 30 for compute reasons, because as we use transformer, they scale quadratically with the sequence length, so you don't want to go too big. And we showed that if you can get this improvement in performance as the sequence input grows. But also what was interesting for us is that at inference time, if you take a model trained only on 30 kbp sequences, but you actually try on bigger sequences, you keep getting an improvement up to 50 kbp. So here is what it looks like if you do prediction as this 50 kbp resolution, where we predict these 14 elements over a sequence of 50 kbp. So you have 700,000 predictions here. You could use all these probabilities and draw bounding boxes by using thresholds or other heuristic. We thought it would be interesting to show the raw probabilities. And so here you can see, for instance, that the model can capture the genes, the different regulatory elements, and so forth of that sequence. And a final experiment before going to this ChatNT approach was we also tried to see the generalization capabilities of our model to different species. And interestingly, if you take the model trained only on human, it can actually get good transfer on species that are related. For some of them, with almost no degradation. But you can also start training on more and more species, and the more species you add to the mix, the better the performance gets everywhere. Now to kind of conclude this talk, I would like to quickly touch down on a very different idea that we had with the group, and so hopefully one day we're going to mix the two, which was, can we give a voice to these models? So there's been now lots of work that looked at, for instance, models pre-trained in computer vision, and where can we kind of connect them to models trained in NLP, and for instance, get settings where you put an image and you start asking questions. Like you feed the image to the computer vision model, and then you ask questions to the NLP model, and it should be able to answer somehow. So why not doing that with genomics? So in that case, could we take a DNA sequence, put it through the nucleotide transformer, get some representation, and then pass that representation to Llama, for instance, and ask Llama questions. So in that case, we could do all the tasks that I showed previously, but in English. So you could say, is there a promoter in this sequence? Is there an enhancer? Is there a splice site? And you would train the model all the time to answer yes, no, or give you more details. You could even do regression tasks by training the model to output some numbers. There are two main motivations behind doing that. The first was try to make these models as accessible as possible. So now you go from a setting where you need to know how to code, or to import a model in a notebook, to fine-tune it, to analyze the output of the neural net, to transform that into predictions and so forth, where now you just have a chatbot. One day you could even directly discuss with that bot and ask all your questions. But the second main motivation for that work was also to say, can we get actually transfer also at the downstream level? So in what I showed before is that you get some transfer between this pre-training phase and this fine-tuning phase. But in what we've done so far is that we fine-tune on lots of other tasks, but there is zero transfer between all this fine-tuning. You get one fine-tuned model per task. And so if you have 5 tasks about promoters, you're going to get 5 promoters models, but they're not going to learn from each other. And what we showed here is that instead of building, for instance, 18 models for the 18 different tasks of the NT paper, you can build a single model that's going to be able to perform all the tasks by asking the questions in English. But not only you can retrieve the same performance with that single model and being interactive and so forth, but you can also get transfer. And what was interesting for us is that if you look at all these tasks on the right, they actually correspond to like detecting histone marks in the model. So in all these questions, there is the name histone that appears. And we showed that somehow the model managed to connect this concept of the histone world in what is actually an histone in the model. And thanks to that, we managed to improve even further the performance of the model while being interactive. We didn't stop with this 18 tasks. We also wanted to see if we can get further. And so we increased like, the pool to 30 tasks. So actually, the 30 do not include all the 18 for the first round. We can just really try to cover as much diversity and species as we can. We also wanted to look in that idea of building like models that can understand all the biology. We also looked at RNA tasks, also at protein tasks, making the assumption that you have the CDS. And we showed that the same model can do classification, but also regression, not only on DNA sequence, but also on protein sequence. So the same model can give the CDS of a protein and ask, for instance, what is going to be the stability, what is going to be the melting point. You expect the model to reply some sentence and give you a number. And what I show is that we did this experiment 10,000 times. Each time we just retrieve the number with a regex, and we plot these values and look at the correlation with experimental data on the test set, and actually the model is very accurate. And kind of, yeah, to finish this talk, I just wanted to show this animation. So it's looking so far, and now we're trying building an interface that looks like ChatGPT, so you can just come and drop a DNA sequence or even several DNA sequences and ask some question that's related to that sequence, and the model should answer, and then you can retrieve the value from that model. So to finish this talk, I would like to thank all the people that made it possible, so at InstaDeep and BioNTech, but also our colleagues from Cornell University, MIT, NVIDIA, and others. Right, thank you for your attention.
All right, probably just one question so that we're not too behind on coffee.
Thank you for the great talk and a lot of great ideas. I just have a question. In an earlier slide, you mentioned that Nucleotide Transformer has learned genomic knowledge without in an unsupervised manner. I was wondering if there's a way to like reverse engineer and see like how the Nucleotide Transformer has distinguished one group of sequences from another sequences.
Yeah, that's a great question. So I would say I see Amber smiling because now it's the kind of questions we're also asking. So I think there are lots of interpretability methods. So we don't have results so far, but yeah, that's kind of on the roadmap of the group trying to use interpretability methods, gradient-based, CNC map to try actually to understand how the network can run these things. Yeah.
That was fantastic. Let's thank Thomas again.